So in this video, we are going to talk about the most important topic, that is Lac Operon. Lac stands for the lactose, operon stands for the operation. Here, it was first discovered by Francois Jacob and Jacques Menard. It is a polycistronic structural gene which is controlled by a promoter and a regulatory gene. Here, the regulatory gene is I gene. Here, there are total of 3 structural genes, that is Z, Y, A. Z stands for the beta-galactosidase, Y stands for the permease, and A stands for the trans-acetylase. Beta-galactosidase function is to break down the lactose into two, that is galactose and glucose, whereas the permease function is to increase the permeability of the cell to the beta-galactosidase. So here we talked about regulatory gene, which is I gene. It is derived from the word inhibitor. It is going to code for a repressor. So now, what's the function of repressor? So basically, the repressor, when there is an absence of inducer, that is lactose, it's going to stick to the operator and will stop the further transcription process. Here the Lac Operon is basically for the bacteria to gain. The question arises that why Lac Operon takes place. It is for the bacteria to gain their energy. Basically, Lac Operon is a basic process which is going to metabolize the lactose and will help the bacteria to gain their energy. Here we see the diagram of Lac Operon, which is given in our NCERTs. So here P stands for the promoter, I stands for the inhibitor. Again there is a P which stands for the promoter, O is for the operator, and there are 3 structural genes, that is Z, Y, and A. And when there is an absence of inducer, what is inducer over here? Here inducer is the lactose. So if there is an absence of lactose in the system, that time the I, that is the inhibitor, will code for a repressor mRNA which will go and stick to the operator. Because of that, it will prevent this RNA polymerase from transcribing the operon. So it will not move further and the mechanism will be off. But when there is a presence of the inducer, there is a requirement for the mechanism to on. That's why the I, that is the inhibitor, will code for an inactive repressor which will not go and stick to this operator. Hence, the whole mechanism will start. So here the Z is going to code for the beta-galactosidase, Y will code for the permease, and A will go and code for the trans-acetylase. So they will create a Lac mRNA where the word Lac stands for the lactose and mRNA we all know. So here first process will be transcription and the second one will be the translation. When there is an absence of inducer in the system, that time the whole process is said to be as a negative regulation.